So we were talking about the glomerular filtration barrier, and we have, this is I think our lecture number 5, and we would be talking about the glomerular basement membrane. The glomerular basement membrane, it contains 3 layers. One is called, so if you see here, an internal layer, a middle layer, and an external layer. The internal layer, if in this diagram you see, this is a layer which faces the endothelial cell. This is called, in electron microscope, this internal layer really looks very light. So it is called lamina. Lamina means a shelf, a leaf. Lamina rara, and rara meaning looking light. Interna meaning on the internal side. So we have a lamina rara interna. Then we have a dense portion. We have a dense portion called lamina densa. And then we have a light portion again on the outer side called lamina rara externa. The outer side lamina in turn faces the podocytes or the epithelial cells. So here these are the epithelial cells. So the basement membrane which is here will be the lamina rara externa. So the glomerular basement membrane is formed of 3 layers: lamina rara interna, lamina densa, and then lamina rara externa. The important thing is for a medical student and a doctor to understand what is the composition of this basement membrane. So first of all, lamina rara interna has collagen 4 in it, which is made up of alpha 1 and alpha 2 chains, and heparan sulfate. So if you remember our previous contextual lectures, heparan sulfate is glucosamine with the hyaluronic acid, and that would impart the negative charge to this. So this lamina rara interna here is strongly negatively charged, and so when the albumin can pass through these big fenestrations, still outside this is the lamina, and that is negatively charged. Then comes the lamina densa, and lamina densa is collagen 4, alpha 3 and alpha 4 chains, and then is the lamina rara externa, which also has the heparan sulfate. So this is the basic structure. The effective pore size of this basement membrane is about 70 nanometer, but it is strongly negatively charged, so it would repel the proteins. So over here, anything larger than 80 nanometer or 100 nanometers cannot cross. Negatively charged ions or proteins will be repelled, not the ions actually, just the proteins will be repelled away. Then once the things come out to the external layer, we have the basement membrane, which again has strong negative charge on it, and it allows only about 70 to 80 nanometer size things to come out, but again it would repel the negatively charged molecules, for example proteins. So this is the second layer of resistance which is present in the glomerular filtration barrier. Then the final or the outermost barrier is the epithelial podocytes. So podocytes, the important thing to understand is that if you see here, podocytes have 2 types of foot processes. So these have long feet, which are called primary foot processes, and then there are small finger-like projections from these foot processes, and these are called secondary foot processes. So if you see 2 podocytes over here, you see that between their processes there are gaps which are called slit pores, and as we talked before, podocytes are really covered with the glycosaminoglycans, and that causes their slit pores to stay open, because these fingers, they try to slip past each other because of negative charge. But these are bound to each other via the nephrine molecules, which are also negatively charged. So the size here for the slit pore is about 20 nanometers, but then when you combine it with the negativity over here, the effective pore size is about 8 nanometer. So if you see here, we have first barrier at this time. This is size barrier and charge barrier, 100 nanometer and negativity. Then the second barrier is the basement membrane, again size and charge, and so about 70 nanometer and negative things are repelled for example proteins, and finally the podocyte and the slit pores, which have the diaphragm on it with the nephrin, and the effective size comes down to 8 nanometers. We will continue in the next lecture.